Section C for the 2017 DEB mock, the answers are as follows. So it said all organisms have the ability to maintain constant conditions in their cells, such as fluid balance, temperature, and chemistry. What term is used to describe that? So the term is homeostasis. Name 2 organs other than the kidneys which help the humans maintain constant fluid balance. That would be your skin, as in you sweat, right, which gets rid of water, and your lungs. When you breathe out, you also get rid of water vapor. Right. Then part B, it says urine production involves the process of filtration and reabsorption. Explain each of the following processes. So filtration means that small substances are pushed out of your bloodstream into your kidneys, and that happens in your Bowman's capsule and glomerulus. And reabsorption, nutrients are returned or taken back into your blood, and that will happening along the loop of Henle and your proximal and your distal convoluted tubules. Right, the nephron is the functional unit of your kidney. Name the site of filtration, so I've just answered that, so your glomerulus and your Bowman's capsule. By what process is glucose reabsorbed in the nephron? It's diffusion. So diffusion is just simply the molecules are moving from where you've lots to where you've less. Give the precise site of action of the hormone ADH, which stands for antidiuretic hormone or vasopressin in your nephron. The precise site of action is your collecting duct and or your distal convoluted tubule. Right. It says a medical lab tested 2 samples of urine. The first was found to contain protein. Would that be considered normal? The answer is no. And the reason is protein molecules are generally too big to pass into your filtrate. However, if you're pregnant, it can be common enough for that to happen. Right, and then they'll check you out and do various different things to you. All right, second urine sample was found to contain glucose. Would you consider that to be normal? The also correct answer for that one is no. And the reason is glucose should have been reabsorbed. So if you have glucose in your urine, you will be a diabetic. Right. Part C, it says give a precise location in the human body for each of the following endocrine glands: your thyroid, your adrenal, and your thymus. So your thyroid is found in your neck. It's found where your Adam's apple would be. Right, your adrenal glands are found on top of your kidneys and they look like little hats. And your thymus is found behind your breastbone. It's a butterfly-shaped gland. Right. Name the hormone produced by each of the glands and give a function for each of those hormones. So your thyroid makes thyroxine and it controls the rate of metabolism. Your adrenal glands make adrenaline, which causes your fight or flight response. And your thymus makes thymosin, which matures or activates your lymphocytes. So it basically works as part of your immune or your defense system. Right, part C, it says an insulin injection was administered to an individual at 0 minutes. The graph shows this injection of insulin affected the blood glucose levels of that person. So what's the effect of injecting the insulin? It reduces the percentage of the blood glucose. Right, so when you look at the graph, it dropped from 100 to 40-ish, so it dropped. Right, how long did the insulin injection affect the glucose in your blood? So if you have a look, it started off at 100, sorry, and it still isn't up to 100 by 90 minutes, so it can affect the level of glucose for more than 90 minutes. All right, it's not just here because this level is not back to the original level. Okay, and what property of insulin means it has to be administered by an injection? It's a protein, so if you ate it, as in it's a hormone, it's a protein, if you ate it, your stomach would digest a protein, so that's why it has to be injected. Okay, question 11, what's meant by genetic engineering? So it's a term you had to learn off. It's the artificial manipulation or alteration of genes. State one application of genetic engineering in each of the following. So a plant, you could say you make herbicide-resistant plants, you can make slow-ripening tomatoes, you could make golden rice, which contain carotene from your carrots. You could have said you made rice containing vitamin A from daffodils either. An application involving a microorganism, you can have interferon being made, right? So you have bacteria making interferon. Interferon is a protein made by our T cells in response to viruses. Right? You can have production of human hormones by bacteria, so for example your insulin, and you can have enzymes being made by bacteria. Part B, it says colour blindness is a sex-linked characteristic in humans. Normal vision results from the possession of a dominant gene. What is meant by the term sex-linked? So sex-linked means that the gene controlling the certain characteristic is located on the X chromosome, right? And in particular, it's on the section of the X chromosome that isn't present on the Y chromosome. What term is used to describe a person who has the gene for colour blindness but who doesn't exhibit it? They are known as a carrier. Then the fun part, it wants you to show by means of diagrams the genotypes and phenotypes of the possible offspring of the cross between a colourblind father and a mum who is heterozygous for the condition. So in terms of their description, they are using your X's and your Y's and your letters beside it. If I was doing it, and I can't really show you by talking through the marking scheme, I would draw sticks, so matchsticks. So you have a big matchstick being your X, and on it you have the little c, and then you have a small matchstick being your Y, and on it you have nothing. That's your dad. The mum has two matchsticks which are big because she is XX, and on it she has a big C on one and a little c on the other. So the gametes they can make, you separate the matchsticks out. So they could either have, from the dad, they can have an X chromosome with the letter C on it, or they can have the Y chromosome with nothing. For the mum, you get an X with the big C or an X with the little c. Okay, you carry out the cross, so you basically match up the gametes. Okay, and you would have XC from the dad with X and a big C from the mum, or XC from the dad and X and the little c from the mum. Right? Or if the dad is giving you the Y chromosome, you would have Y with nothing from the dad and the X and the big C from the mum, or Y and nothing from the dad and X and the little c from the mum. So in terms of the phenotypes, phenotypes are what the person looks like or what they express. So they have females with normal vision, females that are colourblind, males that are colourblind, and males that have normal vision. Okay, but if they ask you a question to do it sex-linked, draw the matchsticks, it's so much easier. Right, with those results you had to say what percentage of the male offspring were colourblind, so you got 50% of them. Is it possible for this couple to have a colourblind daughter? The answer is yes. And explain your answer. So both parents carry the gene for colourblindness. Right, so it's not just the daddy giving it. So the mum could give an X chromosome with colourblindness. And then part C, it says, in your answer book, state which of the following statements are true or false and give a reason. Right, so it says messenger RNA is found in the nucleus and the cytoplasm of the cell. That is true. Reason being, it's made in the nucleus and it moves to the cytoplasm. It brings the DNA code to ribosomes. Down syndrome is a condition caused by a gene mutation. That is false. Down syndrome is caused by a change in the chromosome number. The person will basically have an extra copy of a particular chromosome. So instead of having 23 pairs or 46 chromosomes, the person will have 47 chromosomes. Right? The shape of a protein in an enzyme is vital to its function. That is true. In terms of proteins, you can either be fibrous or globular. Right? So the globular ones are your enzymes, and they have to have a particular dent in it, which has to be a particular site shape, so that the substrates can fit to it. Okay, transcription occurs in the ribosomes of the cell. That is false. Transcription is the making of the messenger RNA from the DNA. So that has to take place in the nucleus because your DNA is only found in the nucleus. Translation happens in the ribosomes. Question 12, it says humans can be classified as primary consumers or secondary consumers. Explain each of the terms. Primary consumers are organisms which feed on plants or producers. And secondary consumers are organisms which feed on animals, or primary consumers or carnivores. So think about it. Humans, by our nature, we are omnivores, so we eat both meat and plants. So we can be both primary and secondary consumer. Give an example of a producer from a named ecosystem you've studied. So you have to name your habitat and name a plant that grew there. All right. So think of the slides and what you can find outside in a grassland. So you can find grass, buttercups, daisies, dandelions. The list can go on and on and on. What's the function of the nitrogen cycle? It makes inactive nitrogen from the air available for use by living plants and animals. Part 2, it says microorganisms in the form of bacteria are important drivers in the nitrogen cycle. List any 3 categories of the bacteria and just briefly describe their role. So the categories are, you've got nitrogen fixing bacteria. They basically convert nitrogen gas to nitrates, right? So the nitrate is the ATE version, which is the form that is available to use by plants. Decomposers or bacteria of decay break down your plant and your animal material, plant and animal protein, basically to ammonia, which is released into your soil. All right, then you have nitrifying bacteria, which convert ammonia into nitrite, and then it can convert them on into a nitrate. And then denitrifying bacteria will convert nitrates from the soil back into nitrogen gas again. Okay, it says name another type of microorganism which plays a major role in your nitrogen cycle. So another type of microorganism is a fungi, and its job is it is a member of decay as well. So it converts dead plants and animals back into ammonia. Right? During the nitrogen cycle, plants absorb compound X from the soil. It's used for the synthesis of plant proteins. Animals break down the proteins to form compound Y. What are X and Y? So X is a nitrate, so it's NIT or ATE, and Y is an amino acid. Don't fall for Y being urea, because it is in the sentence it said it's used in the synthesis of your animal proteins. Urea isn't, okay, but amino acids could be. Part C, it shows you a graph of the population of moose in Canada. It says moose don't have a natural predator and wolves arrived in around 1950. And it says moose eventually became a stable part of the wolves' diet. So it's showing you the graph. So in 1900, we had no moose. By 1930s, you had loads and loads of moose. You had over 4,000 of them. 1950, they dropped to below 1,000. And then they rose up again to about 2,000 by 1960. So it says, why, other than because the absence of predators, in your opinion, did moose population increase rapidly? They could have increased rapidly because they had loads of food, they had a lack of competition, it was good weather. All right, suggest a reason why the moose population decreased between 1930 and 1940. Could have decreased because humans went shooting them, all right? So they were cold. It could have decreased because there was a lack of food, there was change in weather conditions. So basically they weren't happy, they weren't breathing as much. Okay, what effect did the arrival of the wolves have on the moose population? So the moose population initially increased, but then it decreased. Right, so basically the wolves started eating the moose, so it reduced their numbers. Suggest a reason why the moose population leveled out from about 1970s. It says basically that the moose population was a size that could be adequately supported by your environment. There could have been survival of the fittest, there could have been conservation, there could have been less wolves. Okay, just write a sentence. Suggest one possible effect of the moose population and one possible effect on the wolf population if a niche was created for another herbivore. So a niche is a functional role within your environment. So if there was another herbivore, so plant eater, introduced, what would happen? So moose population, it could decrease because the herbivore could be eating its food, or it could stay the same if the herbivore was eating a different form of food. Wolf population, it could increase because it now has more food. It can now eat a moose or the other thing. And it says give a reason for each of your answers. So they are discussed. Okay. Question 13. It says enzymes play an important role in controlling metabolic reactions in plants and animals. What's an enzyme? An enzyme is a biological catalyst. Comment on the shape of your enzyme. It's a 3D folded globular protein. Name the process other than digestion which occurs in a plant or animal cell which requires the use of an enzyme. So think there's loads of them. Germination, photosynthesis, respiration, loads. Okay, describe with the aid of diagrams the active site theory of enzyme action even. Alright, so if you look at the pictures, your enzyme looks like a capital E, the substrate looks like a dumbbell. It doesn't quite match, but it joins up and makes your enzyme substrate complex. When it matches up, the substrate changes its shape slightly. No, substrate stays the same shape. The active site changes its shape slightly. It gets processed, the substrate gets released, so it's now known as a product, and your enzyme remains unchanged. Right? What's meant by the term denaturing in relation to enzymes? Denaturing means loss of shape, loss of function. So if you think of your enzyme being like a big blob of wax, if you heated up the wax, it would melt, it would shape, its shape would change. Right? State 2 ways in which an enzyme can be denatured. You can heat them, you can put them in salt, you can agitate them, you can put them in alcohol and melt them, or you could change their pH. Name 2 enzymes found in the digestive system and state precisely where each is active. So amylase is active in your mouth, amylase is also active in your duodenum, lipase is active in your duodenum, and pepsin is active in your stomach. Last part of question 13, it says bioprocessing often involves the use of immobilized enzymes in a bioreactor. What does the term immobilization mean? So immobilization means attaching or fixing the enzymes to each other or to another inert material or trapping it in a gel. What's a bioreactor? A bioreactor is a vessel, basically a container, in which living cells or their products are used to make a product. Right? In the course of your practical studies, you prepared an enzyme immobilization. Right? So name the enzyme you immobilized. You immobilized sucrase. Right? So we immobilized yeast, but yeast contains sucrase. Name the substrate you use. The substrate you use is sucrose. And name the product reaction, it's glucose. So that's the one where you put your gel beads into dropping funnels, ran sucrose solution through it, checked with Benedict solution and saw that you got glucose. So how did you test for the presence of the product? You use glucose test strips or your Benedict solution. How would you store immobilized beads overnight? You would rinse them and cover them with water, right? Or you could dry them and stick them in a fridge. And gave 2 advantages of using immobilized enzymes rather than free yeast. It can be reused easier. They're easier to extract. You get pure products. They get increased stability. Okay? So that was your question 13. I will continue with question 14 and 15 in another presentation.